And then, of course, I want to invite everybody. I know that we're limited with our setup, and so I invite all of you to sit close together and to move forward and to act like you like each other, so that you have a better view and so that perhaps it's a little bit better. If you happen to be you see it perfectly from back there, then you're awesome. And then one more housekeeping announcement, and it's not really housekeeping, but for you guys, at lunchtime, and we'll try to remind you at lunchtime, today, across the street in the parking lot of the Witten building, which is the building that's connected to this building by the little bridges and by a tunnel, in the parking lot, and you can see it when you walk out of out of Wing 5, you can see it across the street. It's the last USDA Farmers Market of the season. So it's a pretty cool market anyway, but they're doing a lot of extra special fall pumpkin stuff and some pumpkin soup sampling and lessons, and there's also some different food vendors that set up to make things while you wait. So if that's something you're interested in at lunchtime, it's a really cool experience, and since you have an hour and a half, you have plenty of time to go around and enjoy all the local vendors. So that'll be a pretty cool opportunity. And it's the last one. So with that, I want to welcome you guys to the second day of the WGS public meeting. Thank you for all being here. Thank you for taking your time to be with us. This is really going to help us as we go forward to have gone through all these perspectives, to have this dialogue, and to inform our decisions going forward. Today we're going to hear perspectives from the international and national partners and stakeholders. WGS is obviously being employed in many countries and international organizations, including the Global Microbial Identifier, GMI, the World Health Organization, the International Standards Organization, which most of you know as ISO. And everyone is attempting to develop international consensus in this growing field. We've asked representatives from GMI and two major trade partners, Canada and Mexico, to be here today to provide their perspectives on the use of WGS in public health. The speakers are Jorgen Slundt from GMI, Kathy Carrillo from the Canadian Food Inspection Agency, and Brenda Martinez, who will be speaking for Mexico's National Service for Health Safety and Agri-Food Quality, or SENASICA. And again today, to keep up with our ambitious schedule, we will be alerting presenters with our handy-dandy colored sheets of paper with 10 minutes, 5-minute warning, a 1-minute warning, and then a little card to tell you when your time is up. And that's just to keep us on schedule because we've crammed so many wonderful speakers into our day. And then please hold your questions for the end of the presentation after these three speakers. As we've done yesterday, we'll have a panel quick Q&A afterward, and then we'll take questions from both in the room and from online. The last warning is, of course, if you're on the phone listening, please mute your phone as we're ready to begin. And with that, I'm going to turn it over to Dr. Jorgen Slundt.
Good morning, everyone. It's really, really early. I'm flying in from Singapore, but originally I'm from Europe, and in Europe we don't start this early. Generally we are really lazy in Europe compared to the U.S. So sometimes it's good to come to the U.S. and then see how things are done in some areas. So I'm going to talk about this breakthrough that I believe that we are seeing just now, which I think is the biggest thing that has happened in microbiology for more than 100 years. And I'm not really joking. So next. Yeah, there's going to be a lot of that. So I'll rely on you to move on like this. But here are some of the things that happened in the good old days. That was when Europe was leading in science. Now no more. Well, maybe they are in some areas, but certainly not in all. And if you click next one, I think this is the big thing, and actually the biggest thing since Pasteur. And since Pasteur and Koch started to make pure cultures of microorganisms. Basically, we are identifying microorganisms the same way that Pasteur did in from 1872 or 76 or something like that. So nothing really big has happened since then. I know that we also had some serotyping and things like that, but nothing really big has happened since then. We're still using the biochemical testing to identify microorganisms. We've really been slow in microbiology compared to many other areas of science. But now, next generation sequencing will take us to the next level, in my opinion, especially if we get our act together. And my point is that we are not really getting our act together at the moment. So next. Yeah, next. One more. So these are three statements, and you can just put them up like that, that came out of a meeting. One more, yeah, and one more. We're saying that we now have the possibility to share these DNA data across the internet in almost real time, and this gives us fantastic new opportunities. And this is a statement from what we now call the first GMI meeting. GMI was not really formed at that time, but that was the first international meeting when we got people together from all over the world, and everybody agreed that this is something that could be done. So that was in 2011. Next, please. This is a statement from a smart Mexican guy who worked in Mexico, was Minister of Health in Mexico, was Assistant Director General in WHO, and now Dean Howard School of... I've heard that he's moved on. Maybe he's in Singapore, I don't know. All the good people always end up in Singapore, I think. Or maybe the old, worn-out people end up in Singapore. It's more like that. So the advantages that we would see in relation to this, I think I'm listing 4, and you can just put them up like that. So we will be able to diagnose, identify, characterize microorganisms in a totally new way. Outbreak investigation, we talked about that several times yesterday. Prevention and surveillance, we have a totally new opportunity in relation to that, also at the global level. And then research in general. These are the 4 that I'm listing. Next. So we should not only think in these areas, because there are a number of other areas where we are also clearly gaining some new momentum by actually using this technology. And the clinical area, I'll get back to that a little bit later on, because the detection possibility that we will have is again totally new. Metagenomics, we already now have global projects where sewage is analyzed in a small, unimportant country in north of Europe from more than 80 major cities of the world, and in there you can then find basically everything by metagenomic testing. Imagine what that would mean for global surveillance if it was put into a real global system. It's a really fantastic opportunity. Basically, you just scoop up 2 liters of sewage from wherever you live, you freeze it to -80, and you send it to Copenhagen. I mean, you could also send it somewhere else, but that's where they're testing it. And then they do metagenomic testing, and then they put out the results, I hope. I'm not quite sure, because Denmark is now trying to keep data, to themselves. I don't know. You don't know anything like that, do you? Keeping data to themselves? No. That would really be stupid. But of course, we know Denmark is sometimes stupid. Linking human and animal pathogens is also really a big thing. Outbreaks, talked about that. Source attribution was also already mentioned yesterday, and I'm going to also have an example here. Next, please. And then we don't talk so much about this economical issue, at least not in Europe. I know that sometimes in the US there are some estimations, and basically they are very, very crude estimations. But just look at this one. We're talking antimicrobial resistance, which most likely, if we don't do anything, will increase tenfold, at least tenfold at the global level by 2050, when we believe that 10 million people will die every year in the world from antimicrobial resistant microorganisms. Just imagine the cost there. This is the cost at this time, so 0.1% of the GDP in the US. So 10 times that is, I think, 1% of the GDP. And this is only AMR. So we're talking about a huge, huge cost. And of course, a lot of dead people. I mean, 10 million per year in the world is more than cancer. It's more than cancer. Cancer is 7 million in the world per year. This is according to WHO. We have to believe the data from WHO. Next, please. So we're getting into a system where we have a totally new way of forensics. You all know that. We talked about that yesterday. We can use it for all the different types of microorganisms that we have. We can use the data immediately, almost immediately, if we want to. It's routine in the US. Again, I would say the US is ahead of the rest of the world in relation to that. And you can do traceback investigations in totally new ways, something we couldn't do before. Next, please. So then people say, but why is that so fantastic? So we've had genomic testing for a long time, so PFG, especially CDC, will say that. But that's also because it's a fantastic move forward that, again, came out of the US with PFG. It was fantastic, but that's PFG. We're just cutting the genome into 20 small pieces, and then we compare the size of these pieces. It's really not the same thing. So next, please. Of course, you know that this is whole genome sequencing, and next again. So I would say that if we compare two isolates with PFG, it's like comparing two books by comparing the length of the chapters. So if the length of the chapters in the books are the same, we say the books are the same, which is, of course, totally stupid. Maybe not stupid, but it's not so smart. And it would be smarter to actually compare all the letters, and that is what we're doing with next-generation sequencing, and this is why also PulseNet is moving towards next-generation sequencing. Now, they only take small steps in PulseNet, so they only move to MLST instead of going the full Monty. But that's a long discussion, and we also need to have that at an international level. At some stage, we need to find out why would we not share the full whole genome sequence whenever we are doing whole genome sequencing. Next, please. But that discussion has to be done also at the national level, I think. So this is why we want to do this in relation to outbreaks. So next, so you will see that when we used PFG, this is basically when we could actually document where the outbreak would come from in relation to the food. So we are clearly after the outbreak curve. So we are basically really not helping. Next, please. So the source of contamination only identified after the outbreak. We have many, many examples like that. Next, please. So next. So if we move here, which we could do with next generation sequencing, we would be finding the link before the outbreak. Now, would that be a good thing? I hear clear silence in the room. Oh, there's somebody nodding. Fantastic, thank you. Maybe falling asleep, I don't know. But of course, that would be a good thing. And really, you know, when I hear somebody, and that can be from many different angles of whatever we're doing here, from industry or government, something like that, isn't that really a good thing? Do we really want to do that? Yes, we want to do that. That is why we are doing food safety testing. That is why we want to be able to find the links before things are happening. Industry should love this. Forward-looking industry is loving it already. So we just need to convince the backward-looking industry that this is the way forward. And I believe, you know, at some stage this is going to, 20 years, this is definitely going to be totally pervasive. Everybody is going to do this. So the longer it takes before you actually move onto the train, the worse for your company, in my opinion. But we can discuss that. Next, please. Yeah, so next. So that's just what I said. Next again, yeah. So yesterday we saw something about this, Listeria, cluster metrics. This has been explained. I don't need to explain that again. Clearly, if we had a global database and if we had industry add their data into the global database, that's totally not possible. Of course it is. If we had that, we would actually be finding outbreaks even earlier than what the US is doing now. You have the next one. That's what we've seen now. So clearly, it's documented in the US now that you can find the outbreaks much earlier, and therefore the outbreaks become smaller. Is that a good thing? Of course it's a good thing. You're saving human lives. That's what we're supposed to do. Is it a good thing for industry? Of course it's a good thing. The earlier you stop the outbreak, the more likely it is that the company will survive. And we have many examples in US and in Europe and elsewhere that companies can go down on things like this. So, again, I would say that even from an economical point of view, even from an industrial, food industry economical point of view, it makes sense. Apart from that it also saves some lives, which I think should also count just a little bit. Next, please. So, but we're talking about outbreaks, but I also mentioned that I think that it also concerns other areas. And I think we need to accept this thing that most food safety systems operate in the same way across the world. I know that some countries want to go to the rest of the world and tell them how to do. You know which countries I'm talking about. I'm, of course, talking about Denmark and Sweden and things like that. But we also know that mistakes, no matter how we try to do things in our national setting, mistakes will keep on coming in across the borders, and it does not help to test things at the border. I'm sorry to say that, especially here in USDA and also to US FDA. I think when we look at it in a scientific way, it's really not efficient to test at the border unless you test everything, which of course you would never do. So if you have issues in food with relatively low prevalence, we know, and we know that from plenty of scientific data, that it doesn't make sense to test at the border. It sounds very well to the politicians. We have a problem. Let's test some more. It's really good if we test some more. No, it's not. We need to find a way to work together across the world in order to be able to prevent outbreaks by doing good things also in our food safety system. Thank you. Next. So, this example, source attribution, in my opinion, before whole genome sequencing, we could only do that for Salmonella. If we start employing whole genome sequencing in a smart way, we most likely will be able to do that for all microorganisms. Next. So, the link here is that you link the Salmonella serotype to the serotypes that you have in the different types of food or from tourism or whatever. And in that way, you're able to say what's the percentage of this Salmonella problem that comes from chicken or from eggs or so on. And we have used that. Next, please. And this is just telling you how we're using the serotypes. And because we have more than 2,500 different serotypes of Salmonella, we can do this. And then we can also subtype them further. So it makes it even better. But because we don't really have a same system for other microorganisms, it's not so easy. Next, please. So this was used since, late 80s in Denmark. And so you have the line up here, the incidence per 100,000 humans. And you will see that it started with a top that came from broilers. And they could document that through looking at the serotypes and doing the source attribution. So they started doing something, and you can see up to 91 or something like that, the line was dropping, so number of people getting sick from Salmonella dropping. Then it started to increase again. Of course, politicians would tell the regulatory agency, they curse in Denmark sometimes, but I don't need to say that in English, of course. You made us waste all this money, and now we have an even bigger problem than before. What are you doing? But then they could actually document, no, this is not from poultry, this is from pork. So let's do something in the pig production. They did that. Then there was another problem. Then it was in eggs. They documented that. They did that. Now they have actually taken out salmonella from eggs in Denmark. Salmonella in eggs has been eradicated in Denmark. Just for USDA or anybody else who wants to listen, if you can do something in Denmark, you can do it everywhere. Because Danes are not very smart. Then we have a second, just to prove that, we have the last top there, and they never found out where that come from. They just, Danes are not that smart. But actually, they ended up on a good side. You could argue that maybe you don't need to do anything, the salmonella will drop anyway. Okay, I will not argue that because I don't think it is correct. But actually, they used source attribution to do something sensible, and I think that something like that can also be used for other microorganisms if we introduce whole genome sequencing. Next, please. So this is the global idea. So if we take a patient or a food, we do DNA sequencing of the microorganism, we put it to the cloud, we have all DNA sequences in the cloud, we will get an answer back in 2 minutes, which will tell us what the microorganism is, and whether it's resistant to which and what. We can then treat the patient or the food. I know we don't treat the food, but still, the data can also be used for surveillance if everybody was using that system, because we would upload that to the cloud again. I think there's one more arrow, yeah. So this is basically the idea. If we did that, if we agreed to do that, we would really be helping each other with a machine that can tell us what the microorganism is, how we can treat the microorganism, and with a real-time surveillance system that would be global. So you would immediately know whether something is moving in Hong Kong or Singapore. Not in Singapore, of course. Never in Singapore. There's never a problem in Singapore. Which is clearly not correct, because we have problems in all countries of the world. Yeah, of course we have. But the thing is, this is a fantastic opportunity. Then countries say, no, but is that a good thing? Should everybody know if something is happening in Hong Kong or in Singapore or in Denmark? Is that a good thing? And probably some politicians would say, no, it's really not a good thing. We want to keep it to ourselves. Are they going to keep it to themselves? Of course they are not. The biggest issues with big outbreaks here have been when somebody is trying to hide something. We've had that in a number of cases in Europe. The government of Belgium went down because of a food safety scandal. Not because of the scandal, but because they tried to hide it. The EU Commission was changed. The Commission of the EU was changed because of a food safety event. Not because of the event, but because they tried to hide it. Every time we try to hide anything, we're doing even worse. And then, by the way, we're also doing worse to the people who will die from the foodborne contamination that we're trying to hide if it's spreading. And even in the agreements that we have under the WHO, the World Health Organization, the international health regulation clearly says, and all countries have signed that if you have an event, with a public health event, with an international potential to spread, you have to inform WHO, who then has to inform everybody else. So all countries agree that they have to inform, so there's really no way that you can keep it secret, not even Canada. And Canada knows that because of SARS. And Canada, of course, didn't like that WHO told everybody that SARS was in Canada. But of course WHO had to do that. Of course they had to. And I would argue with any Canadian, including up to the Prime Minister, it doesn't make sense to try to keep something secret. It does not work. Not in today's society. Anyway, it will get out. Next, please. So GMI consists of the platform, community network. You can just go into the web page, and then you can become a member. There's no fee or anything like that. There's no money involved in that. We have 4 work groups. I'll just mention them just later. Next, please. So these are the 4 political challenges. Next one. Repository storage of sequence and metadata, analytical approaches, and ring trials, quality assurance. So just to let you know that under working group 1, so we have a meeting every year, GMI 10, the 10th meeting was in Mexico, May this year. Next one is going to be in Geneva, in the week of 14 May 2018, just before the World Health Assembly. I just want to point out also that Working Group 4 is running these ring trials or proficiency testing for, I think, 60 or 70 labs now. So we are actually we already have a system where we're trying to combine the capacity in different countries in relation to the labs. Next, please. Next, please. So this is just the homepage that you can go into if you want to become a member of Global Microbial Identifier. Next, please. So these are the meetings that we have had since 2011. So we just called them all GMI, even if they might not have been called GMI in the beginning. But number 10 down there in Mexico, and then 11 in Geneva. Next, please. Next. So we know that, that's what I said before, that really all data will get out at some stage, and the worst situation for a government or a company is if it looks like they tried to keep the data hidden. You should never try to do that. And then, of course, there's so much to gain from everyone, for everyone, in relation to sharing their data, and I think that should also be realized, or at least discussed. Thank you. Next. So we don't even, so we are also saying that this will go on in the rich countries. It is already in the US, it is to some degree in some European countries. We have this capacity that if you were in a poor developing country and starting to create your system, why not create it using whole genome sequencing from the start? Because it means that you can use the same lab for everything. Because whatever you want to identify, whether it's a virus or any microorganism, other microorganism, some people don't like to call viruses microorganisms, but I will still do. So whatever, you use the same machine because DNA and RNA is the same, almost the same, and you can detect it and characterize it in the machine, whatever. And again, it's a One Health opportunity. I think that was clearly also mentioned yesterday. Next, please. So we were only able to do something in Denmark because that we got researchers, regulators, and food industry to work together. If this triangle had not worked, it would not have worked in Denmark. In the beginning, of course, the food industry would say, no, it's totally impossible to do something about salmonella in chicken, cannot be done. Chicken, if we want to take them out of these big flocks, chicken will double in price. I think somebody even said triple in price. Now we know that it's maybe 1 to 2% increase in price. It's nothing. It's absolutely nothing. But you have to convince industry in order to get it done. The other thing is, industry might be smart in some areas and in relation to some of these things, because if you really want to have efficient solutions, you need to know something on the ground, and researchers don't necessarily know what's going on the ground. Regulators might not even know too. Of course, they will here in the US, but in some other countries, they might not know the realities on the ground, but industry will know that. So you have a lot of information from industry that you need to move on into the system, and that was why it was successful, I believe, in Denmark. Next, please. So these are the three major obstacles. I would say researchers' pride. What does that mean? So it means that if I'm doing whole genome sequencing of a number of strains, I want it to get into nature, so I won't put it in the open database until in 7 months' time. I'm not even kidding you. This is happening. This is happening. Next, industry's reluctance. I've talked about that. I think forward-looking industry is trying to embrace this already, but we have lots of other industry also. Country sovereign right. I mentioned also that countries think that they are doing good things for their constituents or whatever by hiding data. They are not. They are really not. Next, please. So we have these GMI, I would say, achievements. We have WHO, FAO, and also OIE involved in our steering committee. But we really need to get the discussion into the international level, into the intergovernmental discussion, and that is why we are trying to suggest to countries that this should be discussed in WHO and FAO and OIE, and hopefully some countries will listen. Next, please. So just a few slides from Europe, just so that you don't think we are totally in the dark ages. There is something happening in Europe. They set aside money in the Horizon 2020, which is the 7-year funding program for research in Europe, called Compare, and you can see what it means up there. We got money into that program by saying, US is already doing it. That's the worst thing you can say to the EU Commission. US is already doing that. Then immediately they will put money into this area. Okay, but they are doing lots of good things, and actually along the lines of also discussion of what we're doing in GMI. Next, please. So they want to set it up like that. They are talking about harmonized standards, analytical workflows, and of course we need to have these discussions about how we join forces to do this in the best way, also across the Atlantic, and basically I would say across the globe. Next, please. And they have all these different ways of involving stakeholders, and they're doing all these things the European way. You know, it's always very convoluted in Europe. You have to talk to everyone, and you have to include social scientists, and it's probably very good. Next, please. This just to show you that this is all the major institutes across Europe that are actually participating in Compare. Next, please. Yeah, and this is just an advertisement for Technical University of Denmark, because they have this WHO Collaborating Center, and they are putting out a number of methods or an easy way of actually using your data by sharing openly the different finder, plasmid finder, res finder, and all these other different programs, and they are really easy to use, and this is how it should move forward, in my opinion, at the international level. We need to have discussions across the different countries and the different agencies and the different entities that are working on this, NCBI, DTU, other FDA, so on. We need to get all these people together, and that is actually what is also happening in GMI. But we need some intergovernmental discussion to move it to the next level. It's not enough that we have 250 or 300 scientists or semi-scientists discussing it and agreeing that this is a good idea. We need country representatives to sit down and actually probably at minister level and agree that this is a really good suggestion. Next, please. So we're starting that by sending out a letter to the world, and we are doing that within a couple of weeks, I hope. We had a problem finding the address for the world. I mean, it's like we have to find all these ministries of health, and it's not as easy as you might think. We can't just get them from WHO because they don't want everybody to just send out spam emails. I don't know why. But we will find it. We did it in Wikipedia. Wikipedia is so much more helpful than WHO in relation to this. Okay, next, please. I'm not talking about WikiLeaks. I'm talking about Wikipedia. So this is what we are saying in the letter. Basically, is what I've been saying here, that really if we have this technology, we should actually try to do something like this, and it would help us in a number of different ways, and then just also relating it to the rise of antimicrobial resistance, because that's something that some of the governments, or many of the governments, is now making action plans to do. Now, if they're making action plans just now, why not include whole genome sequencing and how we can share the data across the world for that? Next, please. Yeah, this is just what I've told you about GMI 11, so let's just go through that. So we link it up to the World Health Assembly, and hopefully maybe next year, 2019, they can then put it on the agenda and have that intergovernmental discussion. Next, please. This is just an example so that you would not think that I'm just talking about something I don't know anything about in practice. Actually, I don't really know anything about it in practice, but theoretically, we are doing something also in our region. Next, please. we are actually suggesting to countries around that region to send isolates to us, and then we do the sequencing at the university, and then we look at the results together with the countries. That is a way of showing to some of these countries, this is how easy it could be done. Maybe you should buy a sequencer. So I think you should go ahead like that. I think US should go ahead like that. I know that you have been with some countries. I think European countries should go ahead like that. That is a really easy way to start having that discussion with the other countries. Next, please. Yeah, so this is just the last, I'm happy to say. Okay. Going out with a bang here. So I don't know if, I'm sure that you know this quote, but I've modified a little bit. We choose to create this system in this decade, not because it's easy, but because it's hard. Because that challenge is one that we are willing to accept, because that challenge will bring so much benefit to global health and global food science and safety and global food industry. I forgot to put them there. Thank you.
Thank you. It was not only a great way to set the stage for today, but I think it was the lively talk that we needed this early in the morning, because as you say, we start a little early. So with that, I'd like to welcome Dr. Kathy Carrillo from Canada, and she's going to give us the perspectives and priorities from one of our major trade partners.
Good morning, everyone. I hope you can see me behind this podium. I just wanted to start off by saying I'm a lab person at the Canadian Food Inspection Agency. I don't make decisions. I was brought in about 4 years ago, actually, I started, and I was hired through a food safety modernization initiative. And we were just trying to look at, the CFIA was interested in how whole genome sequencing could contribute to their food testing programs. So I've been working on that for about 4 years now. Next slide. And so I'm going to talk to you today. I'm going to start giving you a little bit of an idea of what the Canadian Food Inspection Agency does in Canada. I think our system is a little bit different than in the US. And then I'm going to talk about how we've implemented foodborne pathogen sequencing in our organization. And I thought it would be a good idea to give you an idea of what value we see in it by presenting a few targeted case studies. And then I'm going to end by talking a little bit about the interpretation of whole genome sequence data, that SNP data interpretation. And this is really only a very small part of the sequencing work we're doing at the CFIA, but I thought for this, at this meeting, this would be the most appropriate. Next slide, please. So the Canadian Food Inspection Agency, we do food testing, but we also do testing for animal health and plant health. So food is part of it. And we also do things like feed analysis as well. And the goal of this testing is to verify compliance to standards and regulations that are set by other organizations, by Health Canada, for example. Next slide. For food microbiology, we have 6 testing labs that are distributed throughout the country. So we are doing thousands of samples of food testing samples every year, and this occurs across the country. And what I want to say about this is that we don't get very many positives. You know, we can do tens of thousands of samples, and we're getting about 250 isolates from this program every year. And that's because we're not testing things like, well, next slide, sorry. We're not testing things like raw meats. That's not done through our organization. So we're generally testing foods that we would expect to be microbiologically safe or absent, not having foodborne pathogens. So we have programs in meats, fish, dairy, egg products, fresh produce, manufactured foods, and we also test food processing environments. We do every year targeted surveys, so we'll take a food commodity and look for, try to get an idea of prevalence in a specific food commodity. But even there, you know, if we've got a 0.1% prevalence rate, you have to test thousands of samples to get 2 or 3 isolates. And I think that's one of the big problems with linking foods to clinical cases. It's going to be very hard to get your food and environmental numbers up. We respond to consumer complaints, and we're also involved in cases of outbreak investigation in resolving, testing foods in an outbreak investigation, looking for scope of the contamination, and we're involved in the hazard mitigation response to the outbreak. We're also members of PulseNet Canada, so as of this year, we're sharing all Listeria sequence data with PulseNet. And they haven't moved to Salmonella yet, but we're starting with the whole genome sequencing in that program. Next slide. So this is really currently the way things are happening in the lab. So of course, we get isolates from the food testing programs, and for the whole genome sequencing, we're only talking about the step where we characterize these small number of isolates that we get every year. Right now in Canada, we have to, we do the biochemical identification of an isolate when it comes in, but these isolates actually, saw that the labs are distributed throughout the country. The isolates have to be sent to a lab in Ottawa for PFG testing, and for serotyping, it goes to another lab in another part of the country. So even just shipping costs for these isolates cost a lot of money for the organization. We wanted to look at how whole genome sequencing could replace some of these things. And so for the past 3 years, we've been sequencing everything that comes into the CFIA. And we're doing this in parallel with the testing methods that are in place, which I think our labs find difficult, as you've heard before. So we've got bioinformatic pipelines in place, so everything that comes in through a sequencer goes through the same bioinformatic pipeline, which works with all organisms. And we do a report of analysis on anything that comes in. One of the interesting things about this, doing this in parallel with the traditional phenotype methods, is that we are seeing cases where the phenotypes are wrong. We had a couple of cases of Salmonella serotyping where we asked the lab to go back and redo the serotype because the whole genome sequence gave a pretty definitive answer, and it was actually the serotype that was wrong. And that's really interesting, that we're getting better results from the sequence data than the traditional methods, and that's going to help us make the arguments to stop doing these older approaches. The other thing I wanted to say was, when we are involved in an outbreak investigation, I've been called for data before we even receive the strains. They're asking me for data. So they want the data yesterday from an outbreak investigation. So we've been really looking at ways of generating the data faster from our machines. Now we're pulling information right off the machine during the sequencing run. And we can do a Salmonella serotyping, for example, based on whole genome sequence within 24 hours of having a colony on a plate. So it can be pretty fast, and you can get the whole genome sequence data very fast if that's important. Next. So this is what we send to our clients. We have a report of genomic analysis, and that sort of summarizes all of virulence factors, sequence types, MLST, RMLST, and if there's any matches to our database. So now we have a database of everything that's been collected since 2009. Then we will have some sort of tree of that to show how closely related they are to a historical isolate. Next. And so we're trying to answer 3 questions. What is it? Is it dangerous? Have we seen it before? And of course, you get a definitive identification with the whole genome sequence. It's, you can really tell what species it is. In terms of is it dangerous, as I think we've heard the past couple of days, that's a pretty hard question to answer. We need a lot more information to understand how virulence factors play into human infectivity. But the question, have we seen it before, is a lot harder than you would think to answer, and that's the idea of matching strains to historical isolates. Next. So one of the things that when we started this, we were asked that, is whole genome sequence data too high in resolution? People didn't want to use it because it was too much information. They didn't know how to interpret it. And I would say that you can look at it at any level. The nice thing about it is you can look at it at any level of resolution. And those CSI shows where they zoom into a license plate and they can read the letters, but you can't actually do that if you don't have those pixels in there. It's the same thing with, whole genome sequence gives you the opportunity to zoom in as much as you want and get every piece of information out of that strain. So if my question in that resolution diagram is the font black, I could tell that from the R. I don't need very much resolution for that. But if I want to know what font it is, I need to look at a few letters and get higher resolution images. Next. And so we can do that with whole genome sequencing by looking at, as you've heard, MLST, which is based on 7 genes, RMLST, next, sorry, which gives you 53 genes that you can look at. And this is a lovely typing scheme because it's useful for all bacterial organisms. Or you can go to core genome, which gives you more information. Next, just the next two. And this, has been really, you can go to the nucleotide level, so you can say, are there any nucleotides different between 2 different strains? Next. So as you've seen in the past couple of days, PFG does not always give you very reliable information. This is a case of, there's a bunch of samples taken from the same factory, and there's 3 different PFG patterns. Next. And you can see, there's only 2 to 11 nucleotides different among these isolates. So based on PFG, you would expect that there'd be more differences among the strains than there actually are. So it's not a very informative technique for these isolates. Next. We had a situation where we had this very unusual Listeria PFG pattern come in off of a cooked chicken product. And at the same time, from another lab, the same very unusual PFG pattern came in from a liquid egg product. And so people were asking, is there a link between these 2 food types? Next. And by whole genome sequencing, we were able to show that there was 274 nucleotides that were different between these isolates. And so that really put a stop to the need to do any investigation in that case. So now we have a really clear answer. Strains are not related, and we don't need to do anything further with these samples. Next. And that happens a lot at, we're asked, because we have our data in our organization, we're not doing a very good job of sharing yet, except Listeria is now going in to the PulseNet system. But we're asked all the time to, provide all the sequence data we have for a certain PFGE pattern when there's a cluster of clinical samples that are being investigated. And I don't think we have one case where we have found anything in our database that matches the cluster of clinical isolates. And so this is a case of Listeria pattern LMACI0015. And we looked at everything we had for 7 years back, and the closest match was 39 to 49 SNPs apart from the clinical isolates. So we could show that, we could exclude all these foods right off the bat, which is fairly useful still. Next slide. But once you do the epidemiological investigation and you get more information from the patients, then they, once you narrow down the food, and in this case it was chocolate milk, then it fits right within the cluster of clinical cases. So here we have 0 to 6 nucleotides difference from the chocolate milk strains compared to the clinical cases. Next slide. This is another case, and this is looking at persistent, when we compare strains to our historical samples, if we do see a match, like a zero nucleotide match, that's pretty strong evidence that it's the same thing. This was Salmonella gaminera in a sprouted flaxseed product. And you can see at the top, there's one that's 715 nucleotides away. That was a Salmonella gaminera from sprouted chia. That was completely unrelated. There's a lot of nucleotides difference, but it was taken in the same time period. So we isolated a bunch of Salmonella gaminera in year 1, and then 1 year later, we got Salmonella gaminera from the same food product, and there was a question of whether it's related, and it was 0 nucleotides apart, so definitely related. And we were able to figure out that the Salmonella was in the seeds that were were being sprouted, but we were not able to isolate any Salmonella from the seeds. There wasn't enough in there to actually isolate any, but when it was sprouted again, the same Salmonella came up and there was 0 nucleotides difference from it. So that provided some very valuable information for the food safety investigators. Next slide. Okay. This is another situation that we see, though. This is Salmonella typhimurium in shell stock. And we keep isolating Salmonella typhimurium from shell stock from the same location, and this is over a 4-year period, and it's always the same typhimurium, but now they're 0 to 72 nucleotides apart. And you see that they're not clustering based on year, they're just all over the place, they're very different. If you saw this in it would be hard to find a cluster of clinical cases associated with this because it's got quite a large amount of variability. Next slide. So whole genome sequencing can be very useful in our experience. We found some really useful information based on doing the whole genome sequencing in parallel with our other methods, but it's complicated. An interpretation of SNP data can be hard because it depends on how the contamination came in and rate of evolution of the strain, as I'll go into right now. Next slide. So we wanted to sort of look at that question about rate of evolution of strains and how that might impact, SNP identification. So we did a field study where we inoculated lettuce with we used 4 different serotypes of E. coli, and inoculated each lettuce head with 1 million E. coli. And then we tried to recover them in, right away at week 1, week 2, week 3. And we did this 2 years in a row and tried to get 3 isolates from 3 heads of lettuce for each serotype, which worked really well at time 0 when you inoculate and you recover right away. But again, after that, it got really, really hard. We got in 2 years in a row, we got none of the 0157 back from the lettuce after week 1, but we got it in week 2 and 3. So we're not sure why this happens. Obviously, we need to do far more samples for this type of analysis. Next slide. But we sequenced all the isolates that we did get, and what was interesting was we saw no nucleotide differences for the 0157 over the period of time of the study. No nucleotide differences for the 0103. We had an 026 in there, but we weren't able to recover it at all, so that was dropped from the study. But for the 0111, we saw a lot of nucleotide variation. Next. And we saw that variation at time 0 of, when we inoculated and then we recovered it right away. So every strain we collected had a median of 5 nucleotide difference from the inoculating strain. We see a little bit of a peak in week 1. We don't know what that is about, but and then we, in general, we're seeing about 5 per strain. Next slide. And if you looked at that on a tree, these strains would look really different. They wouldn't have that. With this particular strain, you wouldn't have that close 0 to 6 cluster that you would expect. And so that's this is a rate of evolution that's higher than what most strains are. And we heard a little bit about rate of evolution yesterday, but there's some evidence that stress might lead to strains to go into a hypermutator state. And that might be something that could happen in a food production environment. So it's something that we really have to think about when and learn more about when we're studying, when we're having to interpret these trees. If you look at the orange box here, you can see this is the same culture that's been sequenced multiple times. So the SNPs are not coming in through a sequencing problem, but it's really a true biological difference. And these were verified by Sanger sequencing. Next slide. So whole genome sequencing is a valuable source of information, and it can be useful in food safety investigation. And in terms of, it gives really good answers. We know that we can distinguish the new strain from a control, which is a question we get asked a lot in a recall. And we can also show that strain has never been in that lab before. But you cannot really right now interpret the whole genome sequence data in the absence of epidemiological information, and I don't think we'll be doing that anytime soon. But it can be very useful in targeting your epidemiological investigations. Next. And I'm just going to leave you with this. This is a picture my 7-year-old brought home, and my friends were a little horrified by it. They thought it was a devil with horns sticking out of it. But this came at Christmas. I don't know if you can see, it's actually Rudolph. So context is everything. Next slide. So I just wanted to acknowledge my team, research and development team at CFIA, who is heavily involved in all of this work. We're really lucky we now have 3 bioinformaticians instead of just 1. So we feel like we might get somewhere soon. We have to spend a lot of time arguing with IT over what we need to do this work. And the work on the SNP analysis was done by Austin Markel and Dominic Lambert. Thank you.
Thank you. I think that was also a great look at some of the same concerns that we have throughout different governments and throughout different public health programs, because we're facing the very same thing. So I think you bring a good case for working together. And with that, I would like to introduce one more trade partner. So we have Dr. Brenda Martinez here from, she's going to speak on behalf of Senacica. And I really want to send our appreciation because we threw her in last minute in the deep end because we had some travel issues with our original speakers from Mexico. So thank you so much, and I'll give it over to Brenda.
Thank you very much. Good morning. My name is Brenda Martinez. I am Agricultural Advisor at the Agricultural Office of the Embassy of Mexico. On behalf of Senacica and Zagarpa, I would like to thank the organizers for letting us share Mexico's experience with whole genome sequencing. For those of you not familiar with Senacica, Senacica is a decentralized agency within Mexico's Secretariat of Agriculture, Livestock, Rural Development, Fisheries and Food, responsible for animal and plant health, as well as sanitary regulations of primary production of processing of agricultural, aquacultural, and fisheries foods. It also regulates and promotes the application and certification of risk management systems in order to facilitate domestic and international trade of agricultural products. Within the commitment of keeping Mexico at the forefront of technology and increase the level of trust regarding food safety, Senacica implemented in 2013 the whole genome sequencing for salmonella characterization isolated from fresh and minimally processed products. Back in 2013, only 14 samples were sequenced, but the use of new technologies with increased analytical capacity has allowed exponential increase in sequence samples. As you can see, by 2016, 976 samples were sequenced, and during the first 10 months of 2017, that number has surpassed 1,000. In addition, Senacica has also implemented different pipelines for data analysis and interpretation. Produce as well as animal product samples are used to conduct WGS. The samples are provided not only by Senacica, but also by the Secretariat of Health's Federal Commission for Protection of Sanitary Risks, better known as COFEPRIS, and by academic institutions with whom Senacica has collaborative projects. The WGS general workflow begins with the reception of either the sample or the isolate by Senacica's pathogen detection laboratory. If a sample is received, the laboratory technician proceeds to isolate the pathogen. If the isolate is received, the technician will condition the isolate as needed. Once ready, the isolate is received by the sequencing and bioinformatics unit, which will proceed to extract the DNA and conduct the sequencing and the bioinformatic analysis. Once everything is complete, a report is produced and updated to Senacica's genomic database. This is Since 2013, Senacica has sequenced over 2,000 samples. As you can see in the chart, most of the samples have been taken by COFEPRIS as part of the Salmonella project. Currently, there are 1,872 isolates in Senacica's genomic database, which includes food isolates from fresh and minimally processed produce and animal products, as well as environmental and clinical isolates. Over 80% of those isolates are Salmonella. As mentioned before, back in 2015, Senacica and COFEPRIS developed a joint Salmonella project whose objective is to generate and manage and analyze genomic data to detect and characterize the phylogeographic distribution of Salmonella strains isolated in Mexico. This project is a crucial analytical tool needed to better understand the effects and prevention of foodborne illnesses, as well as AMR gene detection. The isolation is performed by the National Public Health Laboratory Network, which includes laboratories in all 32 Mexican states. The Salmonella project currently has 1,375 sequenced isolates. Most of the samples come from the state of Michoacan, well known for its agricultural production. Most isolates come from processed animal products. As you can see in the pie chart, 58 different isolates have been identified. The top 5 include Salmonella typhimurium, Salmonella natum, Salmonella agona, Salmonella derby, and Salmonella infantis. The pipelines implemented by Senacica allow them to determine AMR gene present in the sequence pathogens, which helps Senacica to collaborate in the National Plan on AMR and strengthen the health and surveillance system to prevent and manage AMR in Mexico. It has also allowed Mexico to cooperate with international partners to strengthen the evidence base and develop new responses to this global threat. The sequencing and bioinformatics unit has also conducted WGS on isolates provided by the National Center for Diagnostics in Animal Health, who leads the National Plan on AMR, and the National Center for Animal Health Assessment Services that provide isolates from the Salmonella Monitoring Program for raw meat in Federal Inspection Time Establishments, also known as TIF. Senacica has 25 sequence isolates to date and has identified 9 different serotypes of Salmonella. As previously mentioned, Senacica has developed different collaboration projects with academic institutions that benefit both parties. For example, the National Autonomous University of Mexico, the largest public university in Latin America, has provided meat isolates and applicants for neurobiological research. The National Polytechnic Institute has provided Idomonas isolates, as well as Salmonella isolates. The Autonomous University of Sinaloa has provided environmental isolates from agricultural water. These joint projects provide students with great fieldwork experience and training using the latest technologies, which has proven very useful and may lead to careers at Senacica or other laboratories in Mexico. With a commitment to maintain the highest standards of cooperation and data sharing to ensure and improve food safety in Mexico and around the world, Senacica belongs and collaborates with networks that have implemented on or are in the process of implementing WGS. Mexico is an active member of PulseNet Latin America and the Caribbean. In November 2015, during the 12th annual PulseNet Latin America and Caribbean meeting, PulseNet Mexico was formed by Senacica, the Institute of Epidemiological Diagnosis and Reference, or INDRA, and COFEPRIS. Furthermore, 3 weeks ago, during the third Genome Tracker International Conference held close by, Senacica expressed its interest in becoming part of the Genome Tracker Consortium. With the use of WGS, Senacica assumes and maintains leadership at the national level as the reference laboratory on WGS, establishes and operates the national WGS network that includes federal public agencies, increases collaboration with academic institutions to develop specific lines of research to meet the needs of agricultural and the agri-food sector, incorporates Mexico into global networks that use WGS to promote and ensure food safety, and strengthen the monitoring and surveillance of animal products through the use of WGS in the national programs of federal inspection-type establishments, which allow to have a genomic type map of the identified pathogens. Mexico is one of the largest agricultural and agri-food producers in the world and a top exporter of fruits and vegetables. For that reason, ensuring food safety and quality are strategic tools needed to increase the competitiveness of Mexico's agricultural and agri-food products. Senacica continues to develop new strategies and partnerships that will allow Mexico to better respond to food safety challenges. Senacica is working with U.S. government agencies as well as stakeholders from industry, trade, agriculture, and academia to enhance food safety. Finally, Senacica collaborates on a wide range of partnerships and has established formal arrangements with U.S. counterparts designed to improve information sharing on emerging food safety threats and to work closely together when addressing product safety issues that may impact consumers on both sides of the border. This is a picture of the team at the highly specialized men and women who work at the pathogen detection laboratory and the sequencing bioinformatics unit in Mexico City. Thank you very much for your attention.
Wonderful, and that's going to bring us to a short Q&A for this module. So if I could have all three of my speakers, I know, we're going to keep you up here, Brenda. If I could have all three of my speakers, we're going to set you up at this table and take a few questions in the room and online. Have we lost Jorgen? Okay, well. And to our speakers, as we answer questions, if you don't mind, when we'll bring up microphones for you guys, but as you answer, if you don't mind reminding the audience of your name, that way our listeners on the phone know who's answering, although I'm pretty sure they'll know if it's Jorgen. Okay, and we'll start. It looks like we maybe have one question in the room. You want to start us off?
Hi, Chris Braden from CDC. One person, two questions, if I'm permitted. First is for Senacica. In your presentation, I didn't see any incorporation of WGS of isolates from clinical cases. So what is the status of conducting whole genome sequencing in Mexico of pathogens from clinical cases, and how is that integrated into your environmental and food databases?
I'm going to have to defer that because I'm actually not an expert. I had to come in from my colleagues in Mexico City, so I can definitely pass along your question, and we'll definitely get back to you via email if we can.
Okay. All right. Thank you. And one other question. So this has to do with the issue of diagnosing clinical cases in humans. The background is that in this last year in the United States, we have demonstrated that culture-independent diagnostic testing of people has actually now influenced our surveillance for pathogens in the United States to the extent that our trend data is actually unreliable, given just relying on our historical case definitions based upon culture. I would like to know if other partners are seeing the same issue, and one of the industry partners that I think is important has not been really addressed here, and that is the diagnostic manufacturing industry. As we look forward, and probably culture-independent diagnostic testing is the future of diagnostic testing in animals and humans, we need to determine what is the platform for testing and what is the data that we're all going to need, and will those diagnostic tests of the future provide the data for public health? So what is the interaction, Jürgen, maybe you can address this kind of more on the global scale, the interaction to address this issue having to do with direct from sample culture-independent diagnostic testing as it relates to the manufacturer of diagnostic tests.
I would always want to speak. Whenever somebody asks me a question, I will speak for the next 5 minutes. But this is clearly a major issue and has also been discussed internationally in GMI, but should be discussed in a broader sense also, I think. So first thing is, if we go to moving away from actually having isolates, we also have this issue that if we find DNA and we sequence it and it fits with a Salmonella enterica type, do we know that this is actually a live microorganism, or was it just DNA from a dead microorganism? Because if you're in industry and you're looking into your products, it's really important that it was alive, because if it was killed, it doesn't mean anything. So there needs to be discussion about how we start using also RNA testing, to combine it so that we can actually have some sort of live dead discussion linked to that DNA sequence that we will use in the future. But but this is a technical issue that could be dealt with if we put some more funding, I guess, into some of that type of research. I know that is going on in a number of different countries now. Again, I would suggest that we would need some sort of international interaction about that, because it's a really, really big issue. I don't know that we have that at the moment. Again, if there was an initiative, an intergovernmental initiative to actually do something actively in this direction, this would be one of the areas that they would cover. Maybe one more sentence, although maybe a long one. Maybe the things that we would suggest to do should be compared to something like CERN, Centre Européen Recherche Nucléaire, which is the big machine under the ground in Geneva where you smash atoms together. This was put together by, 45 countries just after the Second World War, because all countries, maybe apart from the US who had enough money on their own, but all the other countries would say, we don't have money to make a machine like that, but maybe if we got together with all these other rich countries, we could make a global machine, or a machine that we could use for purposes. Now, of course, there's been spent hundreds and billions of dollars on CERN, and I think they found one small particle, and I think we are all very happy with that. But we are suggesting making a major machine that would actually make economies of countries better, that would actually save lives, that would actually do international surveillance, which is something countries have talked about for at least 20 years without moving really forward. And I think, if countries were discussing it in that way, there would be ways of actually creating the big machine, but it would be something the same level or the same size as CERN. And CERN is at the moment 10,000 people working there in Geneva. We could have the same thing in a global machine for something like this, and then we would move forward the agenda in a big way.
So I'd just like to follow up on one thing, so I think it's important to actually talk to the diagnostic development or the diagnostic manufacturing companies, because the diagnostic tests that they're going to produce for the future is probably going to be sequence-based, but the actual data that they use for those tests may be a very small fraction of the data that may be produced. How do we, in a public health community, actually get access to the types of data that we need for public health, more so than just the diagnosis that is needed for patient care? And so I think there is a partnership, a discussion with the diagnostic test manufacturing industry, so that those tests are developed such that that data is available.
There's been some really good surveillance projects recently where they're looking at the sewage projects, and I think that might resolve some of those issues if I think you have to do the surveillance of clinical samples outside, because the diagnostic tests are just going to be cheaper to do, and that's going to drive the movement of diagnostic tests to pathogen-free. So I think we have to have another way of doing this in our countries. And I like the sewage projects because they get away from having to ask permission. And in Canada, we have a lot of rules around permission to sequence isolates from patients. So it gets on around some of these issues.
Yeah, just to let you know that in some countries, they have asked permission to take the sewage and were denied permission. So in Japan, for instance, they had to not send the 2 liters of sewage because Japan, the government, denied them this. I think Japan and maybe one other country, I don't know.
So I know we have one question. We have 2 questions online, and then we'll go over here. So we're going to start with one online. We'll go back and forth. So I think this first question is, and I'm going to give Scott a microphone so he can clarify, is going to relate to actually the last 2 days of meetings, because this question is about, do any of your organizations do random testing at retail, and if contamination is found, is that forwarded immediately for sequencing? And so this is about retail surveillance, or random retail surveillance for pathogens. So that, okay. So if you guys want to comment on systems, I know that this relates to NARM's retail in the United States, but how does that relate to any of your systems?
Yeah, we're definitely taking samples from retail, random samples from retail. So at CFIA, and Health Canada does that as well. And if we do find a positive, then, you know, it goes to a food safety investigation. And everything is sequenced now.
Okay, I'll go over here to the left, and then we'll go back online.
Okay, this is a question for Jorgen. I enjoyed your...
Can you speak up, because one of us up here is really old, you know, and I'm not hearing so well.
Okay, is that a bit better? I'm speaking more directly into the microphone now, so that might help. So enjoyed your talk. Actually enjoyed all three talks. But the question I had was, I was really interested in your point about getting ahead of the outbreak curve. And what I was thinking about that in sitting down. If you're trying to get ahead out of the outbreak curve in terms of human cases, you're only going to have sporadic cases. And it's great with whole genome sequencing that we're able to actually connect some of these sporadic cases because they're actually outbreaks, but there's still a lot of them there. And you mentioned about source attribution, which I think is very helpful. And I think that helps us actually get ahead of the outbreak curve because we can look at the sources of the sporadic cases and perhaps do something about that. But the other thing you mentioned about food safety management systems and what the food industry might do as well. But could you elaborate a bit more on that? Because I didn't really find out what the point you were making on that particular perspective.
So I didn't really get the last part.
Well, so what you said was something about food safety management systems and how the food industry could help us get ahead of the outbreak curve. I think that's what you said in the talk, if I picked you up correctly. I was just wondering if you could express that a bit more clearly, what you were trying to say.
So in the big picture, in the future, somewhere into the future, we will have a big data. I'm not in doubt about that. We're just suggesting we should start doing it now. We will have a big database that will have all the isolates that everybody has at any time in any type of food isolated. We will have that in the database. That means that as soon as we get an outbreak or we have one case, because it could be only one case, we would be able, in principle, to link it to where it comes from. I mean, in principle, yeah? So the idea is that if we had these major databases, we would be able to find issues that we cannot find at the moment, and we would probably find that most of the sporadic cases that we say are sporadic are actually parts of small outbreaks or even large outbreaks that we could never find before, and I think there were even examples of that yesterday. So in my opinion, that would change the whole way of food safety management, away from random testing, which is really not helping. I mean, we've tried that for 30 years, and we haven't done anything to the global foodborne disease burden. Sorry to say, we have not. We have wasted so much money, and I'm one of the guys who have wasted lots of money, so I'm also pointing at myself. We wasted so much money without doing something about a problem. In safety, in traffic safety, they have scientifically looked at where the hotspots are, and they've done something about it, and they've reduced the number of deaths in traffic in X country like Denmark at least threefold over the last 10 years. What have we done in food safety? What have we done? Really nothing.
Well, I think I could probably argue, I think that all our organizations have done some things to defend them. I think the point you're making, that it's really important for our industry when they isolate organisms that they put forward those organisms so that they can be sequenced and put into databases, which will help with the comparisons in the future.
Yeah, but I'm saying we've done nothing because the disease burden has not gone down. I know that in the EU they have taken down the disease burden from Salmonella almost 50% over the last, 10, 15 years. So there they have done something. In the US there is also a reduction at some stage. I don't know if it's continuing or not. But the idea of wasting all this money on all our scientists in relation to food safety is that we're doing something about the problems, yes? I think that's the idea. Or else why would society waste all this money on us anyway? Now we finally have the tool that could actually move forward this agenda in a big way. And then we are still bickering between different countries, between different systems, between different agencies. We have all these problems. We are seeing a fantastic solution. I heard all these presentations yesterday. Fantastic solution that can really bring things forward and could actually reduce the real disease burden significantly. And that's what we're supposed to be here for. And now we're having all these small discussions and we don't want to share the data and whatever. It's really, really depressing when we are looking at this area where we could do so many good things.
So, and I think basically, I mean, you guys are saying the same thing. You know, we have a huge burden globally to attack illness, and we've each been chipping away with it, you know, as we can. But I think, you know, there's a potential working together and with this technology and the great amount of information that it gets us, if we work together, the faster that we get it going, the faster we get this databases in cooperation going, we can take hopefully a big chunk, maybe something measurable, maybe something more significant, you know, globally, instead of these little pieces that we've been doing. So certainly everybody's been chipping away, but we might be able to really ramp that up and accelerate the progress by using all of the tools at hand and actually adopting them faster, which there's always going to be some tape in between, but I think that's why we're all here. So.
Thank you.
And I'm going to take a question from online.
Yes, we have one more. It's, will the global database, GMI, include a surveillance program to sample very high-risk populations like food preparers, farm workers, veterinarians, etc.?
Could you say again?
Yes, will the global database, GMI, include a surveillance program to sample very high-risk populations like food preparers, farm workers, or veterinarians?
Well, so we are presenting the idea from the GMI community, and we are suggesting that it needs to be discussed between governments. So when governments want to discuss something in international setting, like in WHO or FAO, they usually have a background from expert meetings. So we would suggest that we need to have a number of expert meetings which would include discussions about these issues and the whole setup, because this is not an easy thing to do. I'm not suggesting that it can be done in a few months. It'll take a long time, and you need to have intergovernmental discussions of how you want to do that. But these are clearly good ideas, and there are lots of other good ideas to be included in that.
Okay, and I think we've got one more in the room. Oh, Peter, are you going to?
Okay. So this question is primarily for Jørgen, but anybody can answer. In your slides, you talked about the experience in Denmark, where industry and the regulatory work together to use serotyping to identify the sources of salmonellosis and target the right sectors. And so I want to ask a more general question about what were the critical factors that you can take from that, or maybe what's happening in Singapore, to encourage industry and regulators to work together to solve food safety problems?
I'm sure that there were many things, but I'll mention two things. One, that in general, government is perceived to be a good thing in Denmark, which is not the case in all other countries. And I know, if the farmer sees government come to his farm in Denmark, he's happy, because normally the government is helping him or her, which is not the case everywhere. So that's the first thing. The second thing, in general, there was a positive sentiment from industry, food production industry. Of course, they will still complain. I mean, Danish farmers will complain about everything, the weather, also the government and things like that. They will still complain, and they will say, cannot be done, will be very expensive, but in the end they will agree, because they can see science and how it's working. The other thing I wanted to mention is that in the Danish system, there's a lot of collaboration between the different producers. So all the way back from 1870-something, you have this collaborative system. It's not socialism, but you have these, I have to say that in the US, it's not socialism, but it's all the farmers are working together in one big collaborative system. So if they can do that, they can also speak and negotiate in one voice. And I think that meant a lot, because it meant that all the farmers would be doing the same thing. It wouldn't be like, okay, if we do it, what about the other ones? Are they supposed to do the same thing? So I think these were two very important areas. But I think also the tradition of working together in this triangle is also a good. It's a good thing, and it's something that you have to practice. I'm sure you're also doing it in the U.S.
No, absolutely. It was a good question, and also definitely we're going to see that later, because in our stakeholder perspective, we're going to have some of the perspectives from some of the groups of corporations and industry and consumer groups that actually helped make this meeting happen. So we definitely do have that sort of common voice coming through in the United States, and that's exactly why, again, we're here. And so I'll go to this gentleman on the right.
Steve Musser, FDA. Could you, two questions really. Are you using data in the public databases to support your own work? And the second question is, can you talk a little bit about obstacles for uploading and submitting genomic and metadata from food, environmental, and clinical samples in your country, and ways that we might overcome those obstacles to improve the database?
Is that for me?
Well, all three of you are from...
Okay. Yeah, we have, at our organization, there's been a lot of, they asked the lawyers, and the lawyers said no. And that was the biggest problem, and we tried to sit with them and see what exactly their problem was with the metadata upload. It's been really hard to even get a discussion going, but I think right now we just had our first access to information request for genomic data, and it's been a really big, hard thing for our organization to organize. So that might be something that pushes things forward, the fact that, the data we're supposed to be an open data government. So if people want the data, we should have it in a public repository. There's, I think with the metadata sheets that you have, the minimal data that's required should be okay. They don't want identifying information for companies or company addresses or anything like that, but that's not required right now, so I don't think it's anything there. It's about having conversations with the right people and being able to address their concerns in a sort of finding out exactly what the concerns are and being able to respond to them directly. But I find it's always a game of telephone in government, so whoever has the objection, there's 3 levels that I have to go through before I can, and it gets passed back and forth for a long time.
In the region around Singapore, we are trying, with this small project, we are trying to start the discussion, because we will get whole genome sequencing of, 500, 600, 700 strains from the region, maybe more, and then we will suggest to upload them into Genome Tracker or something else, probably Genome Tracker. So then we will start the discussion in that way, and I don't know how it will go, but at least we will start the discussion in 6 different countries, whether that makes sense or not. I know that there are a lot of countries that would say, oh, but this is very difficult, we can't do that. But the thing is, it seems like Genome Tracker is working, even though you are actually putting the isolates up within, as I understand, few days, and I don't see anything falling apart in the U.S., at least not in this area. So I'm pretty sure that it can be done, and again, the example from U.S. that you can actually do that without hurting industry or hurting someone else or hurting other countries would really be a very strong example to take to other countries and say it can be done. But we just have to start doing it. That's also why we're starting that small project there. It's just so that countries will start to get used to that this is the way of the future, and I'm really totally convinced that it's going to happen. Of course, all the concerns of different stakeholders, and they have to be addressed. Again, that's why we're saying we need to have intergovernmental discussion, not conversation between people like us, because we are way too low. Maybe you are not too low, Steve, but the rest of us, we are way too low level to discuss this. This has to be ministerial level in countries. It really has to. And even in some countries, it might have to be prime ministerial level.
Thank you.
Do we have anything else online? Okay, do we have anything else in the room for this panel? Okay, well, that puts us a little bit ahead of schedule, which is never a bad thing because we've got a lot going on after this panel. And so we have a break until 10:30. So that's plenty of time to get coffee, get moving, and then we'll be right back here for our stakeholder perspectives.